This video describes the theory, procedure, and analysis of the drug delivery experiment performed by Group 9, Luke Kinard, Juliette Nealon, and Albert Tulli. An important application of chemical engineering in pharmaceuticals is the rate of drug delivery into the body of the consumer. The rate of drug release is necessary knowledge because ideally the drug should be effective immediately and should provide relief to the patient's problem area. The objective in the experiment is to investigate different variables of drug delivery to find the rate of drug release into the body. To simulate the capsule used in pharmaceutical drugs, hydrogel beads were used. Tartrazine dye was used to simulate the drug being delivered into the body. Regarding the theory for this experiment, equation 1 was used to determine the diffusional properties. F is the fraction of drug released, m sub t is the mass of the drug released at time t, m sub infinity is the mass of the drug released at time infinity, k is a constant accounting for the specific properties of the drug used, t is time, and n is the diffusional exponent. Equation 2 was determined because the volume in the experiment does not change. Therefore, m sub t over m sub infinity can be approximated by concentrations at time t and time infinity, respectively. The natural log of both sides of equation 1 was taken in order to produce equation 3, which will be implemented into the results section in order to calculate the diffusional exponent n and the constant k.
To perform this experiment, 3 adjusted variables were chosen. The first variable chosen was stir rate, and the trials were run at 100 RPM, 260 RPM, and 400 RPM. The next variable chosen was bead size. This was varied by using different size syringe tips when creating the beads. The 3 syringe sizes were 16 gauge, 19 gauge, and 22 gauge. The last variable chosen was temperature. The 3 temperatures chosen were 24 degrees Celsius, 30 degrees Celsius, and 37 degrees Celsius to represent different conditions in the body. Before beginning this experiment, it was important to identify safety concerns that could arise. The most important precaution was to wear proper laboratory attire, including gloves, goggles, and a lab coat. In addition, it was important to dispose of chemicals in the proper waste containers. Any solution containing alginate had to be placed in the hazardous waste container, and any pipette tips, syringes, or similar waste had to be disposed of in the sharps container. Lastly, it was important to use caution when handling glassware and chemicals to ensure no students were injured. The first step in the experiment was to create the solutions required, which includes a tartrazine solution, an alginate solution, and a calcium chloride solution. To create the tartrazine solution, 0.1 grams of tartrazine was added to 1 liter of deionized water. To create the alginate solution, 0.1 grams of alginate was added for every 10 milliliters of the tartrazine solution used. Lastly, to create the calcium chloride solution, 6 weight % of calcium chloride was measured out and added to the tartrazine solution. After making the solutions, they were left on stir plates to mix until the next lab period. This ensured that all alginate and calcium chloride would be dissolved in the solution. The next step in the experiment was to create the beads that would be used to deliver the tartrazine. 30 milliliters of 6 weight % calcium chloride solution and 3 milliliters of alginate solution were measured out. A syringe tip was placed on the end of the syringe, and the alginate solution was then added to the calcium chloride solution by slowly releasing individual drops. Once all the alginate solution was added to the calcium chloride solution, the mixture was left to sit for 5 minutes to allow the beads to cross-link. At the end of the 5 minutes, the solution was poured through filter paper and the beads were rinsed with deionized water to ensure that the beads were separated from the remaining solution. At this point, the beads were ready to use. Once the beads were created, they were added to 100 milliliters of deionized water that was being mixed on a stir plate at a set speed. As soon as the beads were added, a timer was started. For the first 10 minutes, samples were taken every 2 minutes. For the next 20 minutes, samples were taken every 5 minutes. And for the last 30 minutes, samples were taken every 10 minutes. To take a sample, a syringe was placed into the center of the mixture and 2 to 3 milliliters was removed. When doing this, no beads could be collected in the sample. If this occurred, the sample had to be put back into the solution and a new one had to be taken. Once the sample was taken, its absorbance was measured using the spectrophotometer. To do this, the sample was placed in a cuvette and a cap was placed on the cuvette. The sides of the cuvette were then cleaned using a Kim wipe. The cuvette was then placed into the spectrophotometer and a group member pressed run. Each absorbance was taken at a wavelength of 427 nanometers. The absorbance reading was then recorded, and the sample was placed back into the drug-release solution to maintain constant volume. Once the trial was over, the solution was poured into the hazardous waste container since it contained the beads created from the alginate solution. The beaker was then cleaned and left to dry. The syringe used to take the sample was placed in the sharps container, and the cuvettes were cleaned to ensure that no alginate was in them. They were then disposed of in the proper waste container.
To determine the concentration for the collected absorbance values, a calibration curve was created. The calibration curve was produced by making known dilutions of sample and collecting the respective absorbance values. Looking at the linear regression of the absorbance data, we are able to determine the correlation between absorbance readings and their concentrations. Looking further into this regression, it is positive to have a y-intercept value that is close to 0. This suggests that the calibration data is appropriate for all absorbance readings. Here is a plot of the absorbance values for each speed specification. Each point is the mean of 3 individual trials, and the standard deviation is represented for each speed using the appropriate colored error bars. The plot was made for speed, bead size, and temperature. However, for demonstration purposes, only the plots for speed changes will be shown. The dye fraction of each dataset was determined by taking the absorbance values and converting them to their concentrations. From the concentrations, a mass of drug was determined because each test had a sample volume of 100 milliliters. Finally, the mass at any time was divided by the final mass, which represented the infinite mass value. This provided the team with the dye fraction values. Each point on this plot is the average of 3 individual trials, and the error bars represent standard deviations. This plot represents the change in dye fraction as a function of time. As expected, the dye fraction change will approach 0 over the testing period. This is because as more time is allowed for the drug to release, the drug will reach its equilibrium, and no concentration gradient or convection can further promote mass transfer within the system. Plotting the natural log of the dye fraction versus the natural log of time should produce a linear plot for this data. For these regressions, 60% of the data was actually plotted to prevent the values near 0 from influencing the slope and y-intercept values. As discussed earlier in the lab theory, the slope will be equal to the rate control mechanism constant n, and the y-intercept will be the log of the mass transfer coefficient k. From performing a linear regression of this data, the mass transfer parameters were determined and tabulated. Looking at the final mass transfer parameters for each variable, we are able to determine the optimal conditions for the testing to occur. Speed changes had the greatest impact on the mass transfer of the system. Looking at the diffusion mechanism n, the 100 RPM speed is closest to the Fickian diffusion value of 0.43. However, the mass transfer constant k has a value here that is the lowest, suggesting a slow rate of mass transfer. Overall, the optimal condition for the speed is 260 revolutions per minute due to the balance of n and k values throughout the system. For bead size, the k values varied slightly, so the ideal parameter was determined based on the diffusion mechanism closest to Fickian diffusion. Therefore, the optimal bead size is made using a 19-gauge needle and has a diameter of 1.1 millimeters. For the water temperature, 24 degrees centigrade was decided to be the optimal condition for testing based on the mechanism value being closest to Fickian diffusion, and for also having the highest mass transfer rate. Overall, we concluded that the optimal conditions for the drug delivery testing were 260 revolutions per minute, a diameter of 1.1 millimeters, and an overall water temperature of 24 degrees Celsius or room temperature. Thank you.